Hi everyone, it's Sarkan from Tutor Me Well, and welcome to another lecture for BCH311. And in this lecture, our goals are to study the general structures of DNA and RNA and how they're different. And we're also going to look at the nucleotide naming and numbering, so things like how to number bases and sugars. And after that, we'll look at the nucleotide derivatives. Now, these are the bases that are not common in RNA and DNA. And we'll finish off by discussing the importance of thymine in DNA. So why is it that DNA contains thymine but not uracil? So on the right here, you're seeing an outline of double-stranded DNA, okay, in the base pairing, and how it wraps itself around proteins called histones. Okay, you can see these little blobs in here to use less space within the cell, and then that becomes part of your chromosomes. So if you remember, chromosomes are made up of proteins and DNA, okay, and they're found inside the nucleus of the cell, and we can see them microscopically when the cells divide. So moving on, I want to give you a quick overview on DNA and RNA, okay, their structures. So on the left, you're looking at single-stranded RNA molecule, okay, so I'm going to say ssRNA, and on the right, oops, this A is not really A, and on the right, you're looking at the single-stranded DNA molecule. So I want you to pause this video and observe the differences that strike out between single-stranded RNA and single-stranded DNA. Okay, so welcome back. We're going to talk about number one difference between these two guys. And so I'm going to talk from the perspective of DNA. So if you can look at this 2' carbon here, okay, you'll see that it does not have OH. So I'm going to say absence of 2' OH. And difference number two is that deoxyribonucleic acid does not contain any uracil. It instead contains thymine, okay? So this is thymine and this is uracil. And the only difference between those two is that thymine has a 5-methyl group right here. And there's a very good reason as to why DNA contains thymine but no uracil, but we'll come back to that. So for now, I'm going to say no uracil. So both of these nucleic acids are made up of nucleotides, okay? So nucleotides are the smallest building blocks of both RNA and DNA. And so I want to circle one here. So this is one of the nucleotides, okay? And you can circle the rest for yourself. And you can see that this nucleotide, as it's the case with every nucleotide, has a base, nitrogenous base, because it contains a few nitrogen atoms. A sugar, a pentose sugar, we're calling it a pentose sugar because there's 5 carbons in here, 1, 2, 3, 4, and 5. And that pentose sugar is connected to a phosphate group. So I'm going to remove this circle from here because we don't really need it. Okay, and so the next point here is how is it that these nucleotides are connected to each other? Okay, so you can see that this nucleotide is connected to this nucleotide via a phosphodiester bond. All right, phosphodiester. Now, phosphodiester means it's a bond that includes a phosphorus and 2 esters, okay? So you're not going to be asked to show 2 ester bonds, but I'm going to do that nonetheless, okay? So that's one of the ester bonds, and the other one is right here in between oxygen and carbon. So in a way, it's a 3'. I'm going to put 3' in here, and you can see this 5'. In a way, it's a 3'. In a way, it's a 3' to 5'. 3' to 5' phosphodiester bond. By the way, the backbone of both DNA and RNA is made up of phosphorus and the sugar, okay? The only thing that makes the nucleotides different are their bases, okay? Just like, so think about amino acids. What is the only thing that makes them different? Well, it's the side chain, right? Because all amino acids have their core structure. Same thing goes for nucleic acids or nucleotides. Nucleotides have their core structure that we call backbone, and that backbone is made up of phosphates and the sugars. So one useful property of DNA and RNA that we take advantage of in the lab is the fact that they are negatively charged. And you can see where this negative charge comes from. It comes from the phosphate. Every phosphate contains one negative charge. You'll see later on that under application of some voltage, we can actually move both RNA and DNA based on their size. So there's a way of reading the sequence of DNA or RNA. So we'll just look at DNA for convention here. Remember, we always read DNA from 5' to 3' direction. This is just convention. It could have been said the other way around, but it's not. And so what is 5'? You'll see later on that when we number the sugar, 5' carbon becomes this carbon. Okay, so therefore the 5' end of a nucleic acid is the end that has a phosphate group attached to the 5' carbon, and that is free. That is not attached to anything else. Okay, so that's my 5' end, and the 3' end is exactly the same thing. So 3' end has just an OH. Okay, let's assume that's the actual end of the molecule, and there's nothing else. Okay, so it just ends with 3' OH. And so if I were to read this DNA, I would go T, G, C, and A. And I would put the 5' here and 3' here. So 5' thymine, guanine, cytosine, and adenine. Of course, assuming that this DNA was made up of just 4 nucleotides. So you can read this RNA in the same way. You can see that that's 5' end, so I could start from adenine, cytosine, guanine, and uracil, and that's it. And last but not least, before moving on to the next slide, you can see these bonds here. So let me just go switch to RNA. You can see that there's these bonds here, okay, that connects the base to the sugar, all right? And they have a name, of course, everything in biochemistry has a name, and they're called beta glycosidic linkage. Okay, so the base is connected to the sugar with a beta glycosidic linkage. The sugar is connected to the phosphate with a phosphoester bond. All right, and that's pretty much it. So now we're going to move on to the next slide.